Welcome to another lecture in biotechnology. The topic today we are going to discuss is hepatitis B vaccine. Hepatitis B, that virus that causes infection of your liver. It can cause scarring of the organ, mainly liver failure and cancer. It can be fatal. Symptoms for hepatitis B infection. We can get jaundice, light colored poop, fecal matter will be light colored, a fever, fatigue that persists for weeks or months, stomach trouble like loss of appetite, nausea and vomiting, belly pain. These are the symptoms mostly we can see in the patients, those who are infected by hepatitis B virus. What is the cause of this hepatitis B infection? Mainly, this infection is caused by the exchange of body fluids like saliva, blood, and other secretions of the body. And also it is caused by the sharing of the needles from mother to child. We can see the incidence of this infection nearly 20,000 to 2 lakhs per year. This is a diagram of hepatitis B virus. This is a structure of the double-stranded DNA which is present in the virus, and we can see a lot of antigens around it. Mainly the antigen which is prevalent is core antigen, small surface antigen, and E antigen. So these are the prominent antigens present in the hepatitis B virus. We have two types of infection in hepatitis B. One is acute hepatitis B infection. Other one is chronic hepatitis B infection. Acute hepatitis B infection is a very short-term illness that can lead to fever, fatigue, loss of appetite, nausea, vomiting, jaundice, and also the pain in the muscles, joints, and stomach. Whereas chronic hepatitis B infection is a very long-term illness and it occurs in the person without symptoms for a longer period. Coming to the hepatitis B virus, is one of the common virus which infects the liver. It causes liver problems, mainly cirrhosis of liver, and even it causes death. The hepatitis B virus is a 42 nanometer particle containing double-stranded DNA of size nearly 3 kilobase per. This virus is a very peculiar one that cannot be grown in cell culture. So the conventional type of producing vaccine is very difficult from this virus. So only we are going for the different methods using recombinant technology for producing the vaccines. Plasma of infected person contains many antigens. Especially the three important antigens present in the plasma are viral surface antigen HBsAg, the viral core antigen HBcAg, and E antigen HBeAg. The surface antigen, among the three antigens, the surface antigen is present in large quantities and they provide solid and lasting immunity against the infections. The steps involved in the production of hepatitis B vaccine. First, we isolate the hepatitis surface antigen gene from virus by normal process, that is lysine, denaturation, precipitation, centrifugation, and drying. And we are obtaining a plasmid from E. coli using EcoR1. Then we clone this gene, hepatitis surface antigen gene, in yeast plasmid vector to form recombinant DNA. Then this DNA, recombinant DNA, is attached to the promoter, ADH promoter, that is alcohol dehydrogenase promoter, and they inserted in yeast vector PMA 56, which containing a marker gene tryptophan. Then the recombinant plasmid, introduced into the yeast cell, and the cells are selected based on the marker, and mainly they are grown in tryptophan-free medium. The recombinant yeast multiplies in fermentation tank and produces hepatitis surface antigen in large quantities. Then the yeast cell will be ruptured after 48 hours to free these antigens. So this is a diagram you can understand a bit clearly. You can see the virus. This virus is cleaved and removed and cleaved by the resistant nuclease enzymes to remove the foreign gene, especially the hepatitis surface antigen gene. And it is cloned in a vector which is having both bacterial markers and yeast markers and both origin of replications. After it has been cloned in here, the recombinant plasmids is grown in yeast cells, where it is grown and produces lot of antigens that can be sliced and removed and processed and purified. Then it is given as a vaccine. So this is a very important method of producing the vaccine using subunit type. Since the normal conventional method is not available for this type of virus, we are using a recombinant technology method, that is subunit vaccines. This is an example of subunit vaccines where we can produce vaccines. So nowadays, this hepatitis B vaccine has been included in the schedule. It is vaccination schedule. Previously, some 15 to 20 years before, we don't have this type of vaccine program. But now it has been added in all the vaccination program in India.